So, dear colleagues, today I would like to present you my research project revealing the chemical composition of bio-oils derived from spirulina, miscanthus, and sewage sludge-based biomass by soft-ionization high-resolution mass spectrometry. So, I will start with the background. So, comprehensive chemical characterization of the bio-crude is needed to form a basement for the rational design of both production and upgrading step. Implementation of the high-resolution mass spectrometry allows an efficient and complete analysis of the bio-oil components. However, up to now, only limited data is available regarding the influence of the ionization technique, ionization conditions on the observed chemical composition of the bio-crudes. That's why the goal of the project was to understand the influence of the ionization technique, such as electrospray and atmospheric pressure chemical ionization on the observed chemical composition of the bio-crudes derived by the hydrothermal liquefaction of miscanthus, spirulina, and sewage sludge. So we come to the result part. So from the graph, you could see the hydroatom class distribution of the molecule detected in the APCI and AZ mass spectra. For both sources, we could see that the most abundant species was found to belong to the nitrogen-free and oxygen-free molecules, as well as the high share of the N1 and O1 species, which means that this species contains one nitrogen or oxygen molecule, was detected. However, it is worth to mention that for the AZ source, it was found a higher share of the heteroatom-rich molecule, which could be explained by the ionization mechanism of the AZ source. So the chemical composition of the bio-crudes are shown by one Crevillon diagrams, and we could see in general that the AZ source for 3 bio-crude sample show higher hydrogen to carbon ratio as well as the oxygen to carbon ratio. For the APCI source, the higher share of the unsaturated species represented by aromatic compounds and polycyclic aromatic compounds was found. In terms of the molecular weight against double bond equivalent value distribution, we could see that for the miscanthus bio-crude, both sources provide similar data. However, for the sludge and spirulina-based bio-crude, significantly higher share of the species with high molecular weight and DBE values was observed. To understand these differences in the observed chemical composition of the bio-crudes, we implemented principal component analysis. The graph on the left showed the principal component plots, and we could clearly see that the samples were nicely separated among PC1 by the biomass origin and among PC2 by the ionization source used. It's worth to mention that the AZ source, which is represented as the dots, was found to have higher sensitivity toward different biomass types. On the right side of the slide, you could see the loading plots of the principal component analysis. So the loadings is basically weights which are used for the projection of the sample, of the features of the sample on the principal components. And after implementation of the clustering algorithm, we could see that all the molecules could be nicely separated among 4 different clusters, which correspond to the unique group of the biomass and ionization source type. We then analyzed these clusters in terms of the chemical composition, which is shown by the one Crevillon diagram, as well as the double bond equivalent value shown on the right part of the slide. The conclusion of the work is shown in a slide, and generally we could see that AZ source was more specific, addition of the molecule with lower double bond equivalent values and molecular weight than APCI. The PCA analysis showed that we have typically 4 different clusters of compounds, and the further evaluation of the molecular features responsible for this separation is now under consideration. Thank you for your attention.
Thank you very much.